Well, it's my pleasure to introduce today's speaker, Dr. Robert Thacker, also known as SpongeBob. His work focuses on taxonomy, systematics, and evolutionary interactions, and most of his recent work has been on sponges, and especially symbiotic interactions of sponges and communities that they use. He uses it as a model system to look at host-symbiont interactions and the evolution of community structure and mutualistic interactions. So Bob received his undergraduate degree at Duke, poor guy, and then he went to Michigan. I don't know how you all feel about Michigan, but he went to Michigan for his PhD. But then he got to do wonderful postdocs in Guam and Hawaii. He then received an academic position at the University of Alabama in Birmingham and was there for 15 years. He was assistant associate full professor, served as an inter-department chair, and then thankfully we stole him away, and he started this fall as the chair and professor in ecology and evolution. And today he's going to tell us about evolution of community structure in sponge microbiomes.
All right. Well, thanks, Diana. And can y'all hear me? Is this microphone okay? In the back? Okay. What I want to do today is just tell y'all a little bit about different lines of research in my lab. And as the title says, it's going to be about community structure in the microbiomes that live inside of sponges. And on my title slide here, I just put a couple different images so you can see what some of these communities are like. This first one, this is a fluorescent in situ hybridization through a sponge that's a lamellodicidia. But within this, all the bright areas on this slide are where there's bacteria living inside that sponge. Okay? And so you can see that they're scattered all around in the sponge. The sponge in the middle, on the bottom there, that's this Apelsinus fistularis that's super common in the Caribbean, but it gains most of its energy from photosynthesis, right, from the cyanobacterial symbionts that live inside of it. Other sponges also have cyanobacteria that are, say, filamentous. This sponge has a unicellular cyanobacterium in it. The sponge on the top right, this is a sponge larva, and this larva is full of oscillatoria filaments, so cyanobacteria filaments, right? And I think it's just amazing. How can this be an animal larvae that's swimming around, but it's full of all these bacteria, right? And then finally, there's some sponge-bacteria interactions that are more diseases. This is a really common sponge on the lower right here. This is Apelsinus coliformis. It's probably the most abundant sponge in the whole Caribbean. But it has a little lesion on it that might be hard for you to see, a little red lesion there. And that's also a cyanobacterial infection that's a pathogen. Okay, so there's lots of different types of interactions that we can talk about. Make that go. Oops, I might have to do it from here. Yikes. One thing's not working. Is it? Oops, let's see. Yeah, but this one's not.
That's not working.
Yeah, how do we go? Uh-oh. Technology. Let's see. Is it like... Ooh, try this back again. Try that one again. It worked before. Oh, there you go. Okay, that one instead. Okay, we're going to rearrange things. So I'm going to start out with some acknowledgments, too. As Diana mentioned, I moved up here from Alabama, and most of my lab has already graduated and finished up there. So my postdocs and graduate students in my lab have really been contributing to what I'll show you today. But I'm also going to show you some things from two different international collaborations. So we've had tons of people around the world send samples in and help process these and so forth. And I've got just a few of them listed on the slide here. Let's see. But most of the work I'm going to show you today has also been funded by National Science Foundation and the Smithsonian Institution. We can go this way. Oops. Well, we try again. Uh-oh, this one's this way. If we can go there. It's got like a different... Oh, there it went. Yeah, that one. Yeah, okay. We'll try it again. Okay. So these three projects I'm going to talk to you about that have been kind of the big point of my lab the past few years is one is called Porifera Tree of Life, and that allows us to really look at sponge systematics and the evolution and taxonomy of sponges on a really global scale. We've also been collaborating with the Earth Microbiome Project. There's now kind of an official subset of that called Sponge Microbiome Project. where we're looking at large-scale symbiotic interactions between sponges and microbes. And then I'm going to show you how we kind of link these two ideas together today with my third project, and this is called Arbor. And for that, we're building different software for phylogenetic comparative analyses. So if you want to talk software later, we can. I'm not going to show you all nuts and bolts of software today, but that's how we did everything that's in this talk. Okay. Let's see. Oh, if I can get the things to go again. I know, it's the round button, but where do we go? Let's see. This is kind of, it's launching a different thing each time. Here we go. We'll have to try to do it this way. For our sponge biodiversity as well, we can think about how many sponges are out in the world, right? To first get y'all thinking about this. And I don't know how many of y'all go out and look at sponges. You can go see some at West Meadow Creek if you go out there, right, or on your docks around here. But there's just maybe 10 or 20 species around Long Island, okay? If you go out and look at a global census, you'll find about 8,600 named species of sponges. But as sponge biologists, we think there's about double that number, about at least 15,000 that exist. And that's because if you went down to D.C. and looked in the collection at the Smithsonian, you can find just there alone about 1,000 species in jars that just say species A, species 15. They don't really have a name. There's no full description or anything like that on them, okay? And also, we keep adding to this problem every day. Just in one dive in Panama, where I work a lot, in 2010, in one hour-long dive, we found 10 new species. Okay, so you can find lots and lots of biodiversity still in different places in the world. Even places where you think you've really already been and already searched for those. Let's see. Well, I'll try it this way. Okay, and then if we want to think about how we're classifying sponges and how we have sponges and place them in different species, right, in different families, different orders, one way you can do that is just by using your standard morphological taxonomy for this group. And many of y'all may have done this in invertebrate biology lab, right? You can dissolve sponges in bleach or acid and look at all the beautiful spicules that they have, right? And so the spicule content is one way you can differentiate one species from another and try to build up genera and families and so on. But you can also look at how those spicules are arranged. And so for a lot of sponges, there will be a fiber content inside that, and those fibers can be made of chitin or protein. And how those spicules are arranged inside the fibers, are they echinating out of this? Are they located inside the fibers? Are they exterior to the fibers completely? All those are really good characters to use in systematics. Okay? You also have some sponges that don't have spicules, right? They just have these big fibers inside of them. And then some have these fibers and spicules together arranged in a really beautiful way. Okay? And that's one way you can kind of distinguish different groups of sponges. When we look at the global diversity of all these sponges, then, we can think that, first of all, is that evenly distributed or not? This is a map from Rob Van Soest. He did this in 2012. Right? And the darker areas on this map of the world have more species of reported sponges. Okay? And you can see that it's really dark in the Caribbean and up in the Mediterranean. And one way to think about this is the epicenter of kind of sponge taxonomy is in Western Europe, and that's where there's the longest history of work. And then it's also really fun for people to go to the Caribbean on vacation, right, and study some sponges. But that's been where most work has been done. Okay? Other places in the world are not very well explored and well documented, including the whole west coast of North America. Okay? So parts of this coastline in California, you might have like 80 or 90 reported species. But say up around Friday Harbor, Washington, where you have the University of Washington's Marine Lab, there's only about 35 reported species. And I went on a collecting trip a few years ago, and we have at least 100 species from there, right? So there are lots of species that haven't been documented and described around the world. What do these sponges do for you can think, right? In terms of big kind of ecosystem ecology, sponges play a really important role in nutrient cycling, particularly in terms of carbon cycling. And there's a great paper by Jasper the Goose, this was in 2013 in Science, where he documented what he calls the sponge loop. Okay? And the microbial loop is what you might remember from ecosystem studies, is how you get particular organic carbon, dissolved organic carbon, down into your, you can think your benthic fauna, okay, on a reef substrate or out in normal environments. And this might be processing somewhere between 5 and 50 millimoles of carbon per meter square per day. Okay? But the sponge loop that Jasper's documented can process an order of magnitude more carbon that way. Right? You're talking like 90 to 350, depending on what sponge species you're examining. Okay? So in this way, these sponges really could be processing a lot more dissolved organic carbon than were previously thought. And the big question for sponge biologists is this really just driven by microbes living inside the sponge? Okay? Is there just a high density of bacteria inside the sponge removing all this dissolved organic carbon from the water column? Okay, sponges are also really important if you're interested in the big picture of evolution, right? And how do you have metazoa evolution occurring? I've got sponges down here and the bottom there, and remember that this could be the base of your big tree of metazoa. Other studies have found ctenophores to be that base. If y'all want to talk about that controversy with me later, we can talk about it. I'm kind of on this figure came out of our whole portal project and team up with a lot of other tree of life studies. So we're kind of vested in this outcome, but this could be different depending on what types of evidence you have. Okay? But within this group of sponges, the big thing to take out of this is that there's 4 main lineages, right? But your textbook that you might have had for evolutionary biology or for invertebrate biology might have just had 3 lineages for sponges. There's actually 4 really large classes of these sponges. Okay. So with these 4 classes of porifera, you could divide them up a few ways. Okay, you've got these demo sponges, and that's your bath sponges, right? The things that could have fibers or silica spicules in them. You could think about this being about 85% of all known sponges. Okay. Then the glass sponges that are really deep environments are the hexactinellida. Okay, and this is what you would find out in the middle of the abyssal plain would be some glass sponges out there. They can be really beautiful. They have a silica skeleton that has a hexagonal symmetry for it. That's why it's that hexactinellida. Okay. The calcarea or the calcareous sponges, and there might be some of these on docks or in the intertidal zone around here. Okay. And then the homoscleromorpha are this new class. Okay. And that new class is really distinct from all the others because it has more traits that are similar to other types of metazoa. These homoscleromorphs have a greater definition of tissue layers and organ structures, right? They have gap junctions between their cells. All these things that you might think shouldn't be inside of a sponge. Okay, the big change in the past year, in just this past summer, has been to rearrange the demosponges. We've got now 4 different subclasses of demospongi, and these are all new names from what you might have learned before, say, July this year. Okay, so we've had a big changeover in sponge taxonomy, and most of this is the outcome of our portal project, our Tree of Life project. Okay. So I want to show you how we get to those changes a little bit, and what we've done is for this Tree of Life, we wanted to try to focus on this family level for sponges and really figure out how do all the families of sponges interact with each other, right? And so how do we build a larger phylogeny from these families? So we had 142 described families based on morphology, and there's about 700 some genera from those and about 8,000 species. Okay? And we knew that we couldn't spend all of our energy and money at the species level and try to do the same thing for 8,000 taxa, right? We had a limited budget from NSF. So we decided to focus most of our energy on the family level. And so for one representative of each family, we sequenced 7 nuclear housekeeping genes, the whole mitochondrial genome, and then the 18S and 28S nuclear ribosomal cassettes for those. Okay. For the genera, I took one representative of each current genus, and we did the best nuclear housekeeping gene, okay, the one that gave us the most differentiation among families. We picked the best 3 mitochondrial genes, and we still did this whole 18S and 28S cassette. And then at the species level, we sequenced 2 what we call our barcodes. The kind of common barcode region for mitochondrial COX-1 gene, and also a new barcode we invented out of the 28S region. And then for all of these species that we sampled, we also did morphology. Okay, so we've got all kinds of metrics of the spicule types they have, measurements of cell sizes, sizes of their fibers and things like that. Okay, so in my lab, we were in charge of doing the 28S gene component of this. And for all of our sampling, what we did is to grab sponges, divide them up among 10 different labs, and then each lab did one type of metric, right? And so my lab just did these 28S genes. And this gene's about 3,500 base pairs long, and sometimes we can amplify the whole thing as one piece in PCR, right, and just sequence that directly. But usually we couldn't, and we had to amplify 2 long fragments of that gene that would overlap, and then we could stitch them together. But sometimes that doesn't work either, right? Our primers weren't great for everything. And so we could do 2 smaller pieces, but we could always get this 28S barcode region out. Okay. And then I promised a SpongeBob picture, so there you go. Y'all know I really am SpongeBob from that. But we have this whole workflow that we had to try to do to get all these 28S genes. And I really have to thank 2 technicians in my lab, Megan Zappa and Cheryl Carmack, did a lot of this work. And they worked for me for about 3 years just sequencing all these different regions of DNA. For these ribosomal gene sequences, so my lab and also the University of Richmond worked on this 28S. And we had a postdoc at the Smithsonian in Washington, D.C. working on 18S from the exact same specimen. From those, we decided we could align up all this ribosomal data really well if we used the secondary folding structure of the ribosome. Okay? And if you do that, you can actually get a much more accurate alignment. But the problem is you've got to fold this whole thing up. This is just the 18S, and you think the 28S is this thing like twice as big. Okay? So we've got to do all these kind of calculations to fold this correctly, right? And it's just hard to do on the computer, right? It's going to take a lot of computer memory, a lot of time. We also found that we couldn't do this unless we had almost the entire length of the ribosome, right? So that's why we needed this huge, long, like 3,500 base pair piece. But we did see that our accuracy was pretty good if we at least kept things that were, say, 2,000 base pairs long. So the data I'm going to show you today is limited to just this analysis with things that we have more than 2,000 base pairs of DNA. And as I'll come back to, we've still got to figure out what to do with all these short little barcodes and things, right? So we got our optimal folding with this analysis, and we use Phylogenic to do most of this. If y'all have used Mr. Bayes, it's kind of like that, but it builds a lot of different models of evolution along your sequences. The origin of these sequences was kind of neat to see for this analysis. There were 65 sequences in GenBank that we could use that met all our criteria. Christine Morrow is a PhD student at the time in Ireland, and we actually did the sequencing for her degree as part of our Tree of Life project. That's how we got a lot of buy-in from the community. She was already kind of working on morphological taxonomy in this group, and we said, well, send us some sample material, and we'll sequence it for you for free, right? And so we were able to really reach out to a lot of collaborators that way. We had a workshop in Panama where we picked up another 72 taxa that we could include in this and had really fresh material we could sequence from. And then we were able to make a collaboration with the National Cancer Institute, and they provided us with about a third of the specimens I'll show you data from today. That's all from their archive from a drug discovery effort at National Cancer Institute, where they have thousands of sponge specimens, but they had not allowed, say, taxonomists or just other biologists to come in and use those specimens before. And you can see from our taxonomic distribution of these, I have the old labels for those subclasses on here, G1, G2, G3, G4, before we had names this summer, right? And you can see that most of these are in this one G4 group, which is now called the Heteroscleromorpha. Okay. All right, so this group of Heteroscleromorpha is the bulk of our phylogeny. We resolved that really well. Then we also have a separate group here for the order Haplosclerida. Okay. We have a separate group for, used to just be called Varangid sponges. Now they're Varangimorpha. The hexaxonellids, we only have one representative with this long of a sequence. Okay? We have calcareous sponges down here, some hemoscleromorphs, and then these keratosa. But we can resolve all the classes and subclasses of Porifera really well with just our 28S ribosomal RNA sequences. Okay? And there's deep divergences between all of these. So this led to us revising the whole taxonomy of the demo sponges based on all these sequence outcomes. And this was completed this summer with a paper from Christine Morrow. So she just earned her PhD in May, and then just 3 months later, she revised the whole taxonomy of Porifera. So I think that's a great target for a degree effort, right? You can just change the world and how you view an entire phylum, right? And she did this with Paco Cardenas, who was also supported some by our project. He was a graduate student in Sweden at the time. Okay, so we've got several grad students that were able to participate in this effort, and then they kind of led the charge to change the whole classification. We couldn't get all of the all those old fogies to decide on what names should be and how to rearrange stuff and so forth. And so our 2 new colleagues came in and they said, look, this is how it's going to be, and this is how we can revise it. So they put forward this proposal in July, and by August, this thing has been updated. It's in the whole International Code of Zoological Nomenclature now, and we maintain that on a website called the World Porifera Database. So you can go to World Porifera Database, just type that into your Google search, and you can see the whole declassification. So our big question now in my lab, though, is how do we go back to our data and get these thousands of sequences that we have that are short little pieces that we call these barcodes, right? How do we add them back in to this 28S phylogeny? And then we're trying to combine this giant phylogeny with our COX-1 and 18S data set. So that's our big challenge now. It's a lot of kind of computational effort that I'm putting forth on that right now with several colleagues. And we're trying to figure out the best way to reconcile all of that. Okay. The other thing we're doing is trying to map morphological traits like spicules and fiber content and arrangements onto the molecular phylogeny. And so right now we have a target of about 2,000 species with each with 500 traits, but we're building a giant matrix to try to map onto our new phylogenies for this, right? So this is a huge effort as well. We're trying to really go forward on how do our morphological concepts of sponges match up with all this DNA sequencing, okay? All right, so now what I want to do is move into this idea of symbiosis and think about these symbiotic communities that are inside of a sponge as just another trait that we can match up onto that tree, okay? So with our sponge microbiomes, we've got sponges with these really diverse communities of microbes inside them. I showed you this one on my title slide on the right there. Here on the left, this is a nice TEM image that Nicole Webster made. She's a scientist out in Australia and worked with this sponge called Roflaboides. She made a couple of preps of this sponge, put them in the TEM, and got these images where there's no sponge cells, no animal cells in her field of view, right? It's all microbial. So with some sponges, it's more than half the biomass seems to be these really diverse microbial communities, right? So in my lab, we've been working on what forces structure that community. You know, can we think about that in terms of the identity of the host? Does it matter that species A has something different than species B? Does the relatedness of those hosts matter? Can we think about host phylogeny as a force structuring the community? And look at biogeography. How are these different in different areas of the world? Okay. So one of the things that might really help structure this community is the mode in which those symbionts are transmitted, right? And if we can look at this graph for a minute, on the right-hand side, I've got this idea of vertical transmission of symbionts. The middle is the horizontal transmission, and the left is the vertical, right? And then over here on the far right is this leaky vertical transmission. Okay. And what we mean by those is that if you have vertical transmission, you've got some adult sponge here. You know, it's got some kind of mitochondria living in its cells, and then it's also got these kind of darker circles are supposed to be our symbionts in that sponge. If those get packaged up during reproduction into the egg, right, and then they can go on into the next generation, that next sponge would have those same symbionts from the adult, right? And that would be, it's vertically inheriting those symbionts from its parents. But you could also have the case where things can be horizontally transmitted. So what's in the first generation doesn't get into these eggs at all. It just has these mitochondria in there. And then it's acquiring new symbionts from its new environment in the next generation. And so forth for another generation, it's going to acquire different symbionts again. Okay. Then you have this other idea is that things can transmit their symbionts in a mixed mode, where some things could be kind of like a subset of these could be packaged into an egg and transmitted to the next generation. But that new developing sponge could also acquire things from the environment, then transmit a different subset of those to the next generation, and still acquire even more new things. And you can think there's pros and cons to all these different modes of transmission. If you have this vertical transmission, it's also this idea of symbiont assurance, right? The adults are assured that their progeny are going to have the symbionts that they need to survive, you know, when they metamorphose into a new sponge. Okay? You could think for this horizontal transmission, if your offspring are going to a location that's really far away, right? It might be better to have symbionts that are adapted to that separate location, right, or that new location for those offspring. Okay? So in that way, you could have a big advantage from acquiring your symbionts from that surrounding environment. Okay? But for a lot of species, maybe this leaky vertical transmission is better, and it's better to have a mixture of new symbionts coming in, depending on where organisms are found and where the larvae get dispersed during their lifetime. So we wanted to try to investigate this idea on a big scale in my lab, looking at sponges from around the world, and could we see a geographic component or some kind of horizontal transmission component in structuring their microbiomes, right? Or would we only see this idea of inheritance from a parent structuring a microbiome? Okay? And one way we thought we could do this is that if you think adults are always kind of donating the symbionts to their progeny, and that just continues indefinitely, then you should expect to see a lot of differences between individual species, right, and what their symbiote communities are. Okay? If you think everything is acquiring these symbionts from the environment, you can still have a lot of species distinctions, but you might have a lot of instances where similar microbiome communities are observed in similar locations around the world. Okay? So our methods for this were really to work with the sponge microbiome group, and we have about 60 different sponge biologists contributing samples to this effort, so it's a pretty large collaborative project. And in each of our labs, we're extracting metagenomic DNA from these sponges, so it's the sponge DNA plus all these symbionts that makes it this metagenome. Then at just a core set of labs, about 4 different labs, we're amplifying and sequencing the V4 region of 16S ribosomal RNA in these bacteria, and we're doing Illumina-based sequencing on those. And our current data set is about 1,500 specimens, and when you look at the microbial sequences here, we don't really know what species they are for sure, so we call these our operational taxonomic units, or OTUs, right? These are distinct sequences, and these are like 97% similar OTUs. We have about 370,000 unique taxa out of this data set so far. So it's this huge number of types of bacteria. So I'm going to first break you out just part of this thing, right? And what we first started with in my lab as our pilot project for this. We selected 20 species that we had in our freezer already in Panama, from Panama, that we had collected from 4 different locations, and we wanted to get basically 5 replicates of each species. Okay? And so where our work is here is on the Caribbean side of Panama, right next to the border with Costa Rica, and this is called the Bocas del Toro archipelago. There's a great field station there that the Smithsonian Tropical Research Institute runs, and so we have really easy access to all of these field sites. And we have the lab is right outside of Bocas Town here on Isla Colón. Okay? And then these sites are all, say, within about 15 kilometers of each other. Okay? And we included some recognizable sponges like Zestaspongia muta, the big barrel sponge in the Caribbean. And then Apocina fulva is this kind of mustard yellow rope sponge that's very common as well throughout the Caribbean. Okay, then for all of these, we sequenced that V4 region and tried to plot out some different metrics from those OTUs. And for these, what I'm going to show you is first is 90 of those 100 hosts worked, and that's what we have about for our success rate, about 90% getting all the way through our pipeline. I'm going to show you these things where I've done some editing to our data set, right? I set a threshold value of 5 reads per OTU. We think things that we don't see at least 5 times in our data set could be errors, right, and sequencing errors. This reduces our data set for these 90 hosts to, it's like 21,000 OTUs that have an average number of reads of 90 reads, and the most frequent one was seen 77,000 times, right? So when I plot just a rank abundance graph, when I first did that, I thought I was making a lot of mistakes. It just looks like axes, right? This is like the number of times we've recorded some OTU or some bacterium across the number of OTUs that we have. Okay? So you can see a ton of things are really rare, and we just see them a few times. Okay? But many things are super abundant. But it's hard to visualize that, right? You can't really see a pattern there. So I'm going to apply some additional filters for things I show you, as a graph. And some of these are going to just have the 100 most abundant OTUs. And others, I set a threshold of 500 reads, and that's 390 OTUs, right? So here's our top 100, right? And now it looks more like a rank abundance plot that you might be more familiar with. Okay, and then to analyze these, we use this R packages to do this. But we used our new software, Arbor. And so what our Arbor software does, it lets you run any kind of R code or Python code you want, not on your local computer, but at a distributed site. So you can run these on like an XSeed network site. Right now we have a couple servers set up here in New York at a company called Kitware, but we also are putting these on the iPlant site at the University of Texas, so you can run on the Stampede supercomputer there. Okay? So we're going to calculate just a few things from our data set, like diversity of these plots, Bray-Curtis dissimilarity of these microbial communities. And then we'll test this idea, do we have phylogenetic signal? And can we actually see similarity in these communities across our phylogeny? So first we can think about how many of those taxa, or those microbial taxa or OTUs, were present in each host, right? And a lot of times microbial ecologists will rarefy their data set to do that, and we did that here. We rarefied these to 600 reads, and you can see that some things actually have 600 things in them when you hit 600 reads, so this isn't the best thing to do with your data a lot of times. Rarefaction can really distort things. But some taxa don't have that many things in them, and some have maybe just 150 or 200 types of bacteria in them. Okay? When we look at this as a heat map, this is an easier way, I think, to see the same data. Here I've plotted the relative abundance of 100 most abundant OTUs here. And these different big colors here, y'all can't read inside there, but this big one here is like cyanobacteria, then there's gamma proteobacteria. This whole last set here is unclassified. Okay, so things that we couldn't really classify to what type of bacteria it is. Okay. On the left-hand side here, I've got the host phylogeny, and the names of these sponges are over here on the right. But each row then is the community present in one sponge for the 100 most abundant OTUs. And the things you can think from just looking at this pattern is that first, all the hosts are different, and they're all statistically significantly different from each other. And then we see a really different pattern of some of these taxa, like these 3 kinds of Aplycina and Iocoria here. They have, it looks kind of visually similar, which taxa are found inside of which host sponge, right? But there's still enough differences in shading to make these statistically different from each other, okay? But all of them have this trend of having a lot of different OTUs or microbes present in that sponge, but at a pretty even diversity, okay? Whereas other things, this is this Eotrichota down here. You can see this whole row is mostly empty, except for one OTU here is really super dark, and it's 90% of the abundance of bacteria in that sponge, okay? So some sponges are dominated by one type of thing, and others are having a pretty even spread with a lot of different types of bacteria in their community. One way you can kind of quantify that from community ecology is with the inverse Simpson's index. And so I have a little plot to show you how that works, right? If you have like 5 species of trees or something, you're going out and counting, right? And you have 5 of each of those, right, equally divided on this graph, right? Then your Simpson's index here is D equals 5, okay? So the largest that value can be is the number of taxa that you've counted. If your number of species is 5 and one thing dominates this community, 4 things are just seen once, okay, then this D goes down to 1.4. Okay? So what we want to do is calculate that value of D and see how that varies among our sponges, but we want to look for phylogenetic signal in that. Okay? So when we look for phylogenetic signal, you're thinking that things that are more closely related on this phylogeny should have more similar traits. Either the size of these circles is more similar, or the shading that I put on these circles is more similar, things like that. Okay? So we want to figure out, do more closely related sponges actually share more similar communities? And so when we do this with our small data set, we had observed values of D ranging from 1 to 54. And I plotted them here. The size of these circles is proportional to that value of D. And here's our same 20 species of sponges again. And you can see that there's a significant phylogenetic relationship there. There are some clades, like these Aplycinas, that have this really large value of D. You also see that large value down here with this Aerolis and Ectoplasia clade. Other clades have really small values. So all of these sponges, from this Desospongia to Xanthimidon, are in a group called Haploslerida, and all the members of that whole order of sponges have this really low value of D. Okay. So next, once we did this pilot project, we wanted to see, well, can this scale up to our current data set from the Sponge Microbiome Project? Can we get this up to a global scale? And what we found is that we needed the phylogeny of those sponges for this to work. And we either had to find a known sequences from specimens, or we had to generate those sequences ourselves. So we needed either 18S, 28S, or Cochran sequences. We found that we had about 647 tissue samples from 64 species where we could kind of meet all these criteria. Right? And when we look at the communities inside these, we decided we could do some more things on a big global scale. We plotted rarefaction curves, so it's the number of samples that of a particular sponge that we had versus the number of OTUs encountered. Okay? And for things like seawater, this starts to curve over a lot, but for a lot of sponges, we haven't sampled enough to really get a great estimate of the diversity inside of that sponge. Right? When we look at sponges as a group, though, we can see that that's starting to curve over a little bit. And as a group, these sponge samples are much more diverse than our seawater samples that we have. Okay, we can also use some tricks like multi-dimensional scaling to try to see how we can cram this community of 30,000, or 300,000 species into just 2 dimensions, right? And so remember, if you're doing this multi-dimensional scaling, it's like doing principal components. If I have 2 host species, if they have similar communities, I might have a cluster here of species A and a cluster for species B, right? And the distance between those points should be proportional to that dissimilarity, right? And the distance to the centroid, so from each point to the middle of where the cluster of points is, that's proportional to the variance, right, or variability in that community. Okay? So when I give you a messy graph like this, it's hard to see it, right? This is with 67 species in there, and it's kind of a mess. But you can see that there are some tight clusters of things that are recognizable because we have a lot of sampling of some species, right? And when you do a statistical analysis on this, yep, it still holds true. All the species are statistically significantly different communities, okay? But this is really hard for me to interpret as just like an MDS plot. So we also looked at this idea of variability in the communities. And when we do that, we can plot out here our different species here on the left-hand side of this plot. And then what the value is here on the x is the distance of these samples to their group centroid. Right? So we have some things that are extremely variable, right? And they have a really wide difference between each sample of that sponge that you pick up. And then some that are really not variable at all. And each sponge you pick up, each sample, is very similar to the next one. Okay? And then just to show you where seawater sits on here, you might think the seawater would have been the most variable component, our replicate samples of seawater. We actually had several sponge species that were more variable than seawater, right? And that really surprised me. And all those were more variable than just replicate sediment samples that we had. So this really amazed me. And then I started thinking about this in a different way the past few months, and thinking about this idea of variability as this idea of leakiness, right? When you think things can have this leaky transmission of symbionts and be acquiring new symbionts for the environment, that's one way you could generate variability in these symbiont communities, okay? So it could be that these sponge species that are just high on this plot here are just the sponges that are more leaky, and they're letting in more environmental bacteria into their microbiome. Okay? The other thing we've been thinking about in the lab lately is whether this is really a life history trait you could think of, is whether sponges can have a super variable community, and are there advantages to having that variable community, or are there advantages to having a community that has really low variations, really consistent from individual to the next. Okay? And there might not be a single life history solution to balance those 2 opposing forces. And you can kind of see that on here, because this is pretty much a continuous gradient in the means of these species, from being really variable to very low variation. Okay, we can also look for this idea of phylogenetic signal in that diversity, and so we're going to use the Simpson's index again for this. So here's a phylogeny of this bigger set of species. I put the orders of sponges across the top here and tried to box them in. You'll see that there's the one problem from our taxonomy that hasn't been resolved yet is Hylochondrida is actually in 2 places on this, and we're still waiting to revise the whole taxonomy of that order. But within that, you can see there still holds true that we have some clades here that are high D, right? And especially these Vorongids and some of the Dictyoceratids. But these Postuloclarids are all really low D. We actually found some Haploclarids with a high D. Right? Before we had a clade of just low diversity Haploclarids. By adding more global sampling, we find some have more. Right? But this still has a significant value for Blomberg's K, which is one way you measure that phylogenetic signal. Okay? So we reinforced our idea that there's phylogenetic signal, but now what we've been thinking in the lab is this shows that there's natural selection to have this really high diversity community, and is there natural selection for a low diversity community? Right? Is there natural selection for variability in those communities, or natural selection to keep that community really limited and very consistent from individual to individual? Okay, we've also been trying to look at this idea of spatial variability in these microbiomes. And we've done that with our current set of samples, and you can see that we're not really reflecting the whole world yet, right? We don't have any samples from South America and very few from Africa, right? So we need to get more sampling done, okay? But we do see our community dissimilarity is correlated with geographic distance. So things in the same location are more similar to each other, okay? But you've got to be thinking that's also confounded with the identity of the host, okay? So we've also been trying to tease out these separate influences of the host identity, the host relatedness, and this geographic structure. So if we first look at our community dissimilarity, we can find that it's significantly related to both of those, okay, if we just do Mantel test. But we can also do a Mantel test where we hold host identity constant and look at the additional contribution of host relatedness. And it's still significant, but it's a very small correlation there, a very small change in the correlation. We've also been trying to look at geographic distance influencing these communities, given that you know what sponge it is already, and we find a significant correlation versus if you know differences in sponge identities versus their geographic distance, and you hold that constant, it is still a significant correlation. But I don't think that's the right way to treat these data. What we're doing in my lab now is trying to build new phylogenetic general linear mixed models to try to let us incorporate the host identity, the host relatedness, and geographic spread around the globe all at the same time in one equation, and we can get a better test of this. Okay. So to summarize this, our microbiome community structures are really strongly correlated with the host phylogeny, but what's interesting, it's in this idea of the big overall pattern, right? It's this idea that some sponges have a really even community that's broadly variable among individuals, and other sponges seem to have a really tightly controlled community that maybe only has 4 or 5 taxa in that. Okay, so trying to figure out why do you see this big difference among different species of sponges is going to really be the questions in my lab over the next few years. We do know that the community composition and the relative abundance, it's influenced by not just the host identity, but how related things are and where they co-occur. And when we try to model that data, this leaky vertical transmission has been our best fit so far, but we are trying to work on new ways to model symbiotic transmission and evolution from different hosts. Okay, and so I'll leave you with this thing. This is Caribou in Belize, and so we've still got samples to process from there, and that's what that's showing me. I didn't show you any data from there yet, but we have about 500 more specimens from my lab still in that pipeline, representing another 100 species. And from our global set of sponge collaborators, about 5,000 more specimens still being processed. So we have a lot more data coming, and what we're trying to do now is just build a way to handle it all in advance before we get these big data sets. But I'd be happy to answer any questions that y'all have. Yeah, and so for some of these, we have done comparisons of the larval microbiome versus the adult, right? And we can get the eggs basically. One way you can do it is you just put pantyhose over the sponge, and then you can capture the eggs into the pantyhose. It does work, right? And the other thing we do is we have a couple sponges that the one I showed y'all in the first title slide. That I can always get larvae from any day. Like I can go out and break them open because it's always releasing. But what's really wild is probably for about 80% of sponge taxa, no one's ever recorded reproduction, right? So you don't know if they're producing eggs and sperm and just releasing them, or if they're brooding larvae and no one's ever seen brooding. So it's really interesting. The whole group of Varangida, the first larva was seen just last year, right? And so that's also pretty fascinating to me, is that you have the whole phylum where most people have never seen it reproduce, right? That's kind of interesting, right? Yeah.
So I'm wondering how these different diversities of symbionts, microorganisms, actually function in sponges, and what might be the structural patterns within a sponge. Because my impression was you take a whole sponge and you analyze the entire sponge as a homogeneous unit, but within that, presumably these groups of microorganisms are distributed relative to the way that the morphological structure of the sponge is.
Right. Yeah. Right.
Function.
Right. So I wanted to show y'all today some of these big overview things we've been doing lately. For about 5 years before my recent work, we've been working mainly just on cyanobacteria inside of the sponges. And so I can show y'all a lot more about that later. But for about a third of sponges in the Caribbean, they have photosynthetic cyanobacterial symbionts that are providing nutrition and energy to the host, right? So for some sponges, it's about 80% of their energy budget they're deriving from these symbionts, but it's variable because some hosts you can find the cyanobacterial symbionts in, but they're not getting any nutrient transfer from the symbionts, right? So it depends on what species is how it's interacting with the microbiome community there. And so far my lab has only worked with the cyanobacteria in detail for those. But we do have some evidence that other types of nitrogen fixation are going on, like sulfur reduction and sulfur metabolism in some sponges. So there's a lot of different roles that can be played by these. For the sponges that, if I went back to my graph where I have that heat map, sponges that are closely related and dominated by just one type of symbiont, it might be an alpha proteobacterium in the first species you look at, and a delta proteobacterium in its closest relative, right? So they'll have the same type of community structure, but the actual identity of the symbiont is completely different, right? So I think they're doing very different functions, but that's where we still need to get to, right? And somebody,
Yes. Thank you for this great talk. I wanted to ask a question. Do you know the time scale at which they can they swap out their symbionts? Because you were talking about the vertical leaky model, and it made me think that, well, maybe they can expel them and get new ones at different times. Can they do that? Or
right. So I don't have any observations of those. Yeah, I can think of, there's one I've seen where there's one reported study where cyanobacteria were kind of overgrowing a host sponge, and there's observation of the host kind of walling off the cyanobacteria and forcing them out, okay, and kind of trying to maybe expel them, right? But I've only seen one paper with that in it, right? So, but that's a really great idea, and I think that's something that we still haven't explored, is how can you get this kind of turnover in the community, right? And at which point in development is that happening? So we do know that most of the sponge larvae that we've sequenced so far do have bacteria inside the larva, but when they settle and metamorphose, that could be a really big opportunity for things in the surrounding environment to colonize inside the sponge, right? But so we're not really sure yet. Right? Yeah, no one has it. Yeah.
Do you have any indication if the symbionts are involved in speciation?
And
Speciation?
speciation, yeah. And so that's a great question because we're not really sure of all the different kind of speciation mechanisms for sponges, right? For how you have, if it's involved in gamete recognition and things like that. So that's something we could totally explore more, but we haven't seen the actual role they would play in that yet, right? But that's another good idea we can look for. Yeah.
How do you think sponges will fare under climate change, and will it differ for these different large groups that you?
Yeah, I think it differs a lot depending on which group and how their skeletons are based and so forth. So y'all actually have a student that just finished as donor committee, Amber Stubler, that hopefully some of y'all went to her defense and things, right? But with Amber's finding, some sponges that have a more silica-based skeleton didn't really change in terms of their growth rate and metabolic rates in terms of the sponge. But when she looked at some bioeroding sponges, it did increase their rate of bioerosion, right? Because the calcium substrate they're going in, boring into could be more easily eroded in an acidic condition, right? I'm on actually another students committee, Jan Vicente down at University of Maryland. He's doing similar ocean acidification experiments, and he hasn't seen a lot of change in the growth rates of sponges in different pH conditions, right? So it's really interesting that some of the demo sponges in particular might be really resistant to any kind of ocean acidification, but I haven't seen a lot of studies yet with calcarea, and they should be a little bit more susceptible, but I haven't seen that work yet. And then with temperature, it's interesting because for some sponges, it seems that the increased temperature actually increases their metabolic rate, and then doing better overall. Others, it's that it's a more complicated interaction that an increased ambient temperature out in the field increases the frequency of, say, green algal blooms or cyanobacterial blooms, and then those can actually clog a sponge's filtering apparatus and kind of smother it that way, right? So that's a different kind of side effect of increasing the temperature out instead of just doing it in an aquarium. All right. Dave.
So, in regards to explaining the diversity and such wide ranges of diversity, you didn't really put it into a cost-benefit framework or a trade-off framework that we often use. So, surely, if all the microbes were beneficial, then you should have as many as possible. So there's got to be some sort of trade-offs associated with certain kinds of microbes. Some must be detrimental, some must be beneficial.
Right. And so one thing I didn't know if I'd have time, but I actually have a slide for it. Right. So one thing that I've talked about before, I think when I came here to interview and everything, is that for we have a cost and benefit slide, right? So when we work on cyanobacterial symbionts, we worked on this quite a bit, right? And the idea is that for these, we know what our benefit is. We can measure the photosynthesis, we can measure nitrogen fixation, secondary metabolites, and all these things that are produced by the cyanobacteria. But we know that there's also a cost, and for the cyanobacteria, is that the cyanobacteria can overgrow the host sponge, right? And kind of kill the sponge from within just by blocking everything happening, blocking cell-to-cell contact, blocking canals, things like that. So we know that that one's more flexible, and I don't know how well that architecture extends out to other things with, say, a delta proteobacteria, because I'm also trying to find more microbiologists to collaborate to figure out what they're doing, and is the sponge having to limit their proliferation inside the body, right? So that's our big idea for a while, but that's, I think, what we're trying to do now is figure out if we're going to take this global approach and try to look at this large scale, how do we look at these costs and benefits on this big, giant scale as well? Right? Yeah, go ahead.
You brought up this whole concept of sponge limit. Sponges are taking up dissolved and colloidal. But then you mentioned something and how it might be related to the biomass and diversity of bacteria.
Right. So no one's actually established the link explicitly yet for that, right? And so that's what I guess there's at least 5 different research projects I know about going on that are explicitly trying to do that. Is to duplicate Deguche's sponge loop measurements with an analysis of the microbial community and carbon transfer in the bacteria, right? There are a couple papers that have from like the 70s that established that bacteria inside the sponges can be taking up the carbon, but it hasn't been examined on a really big scale and in a really large experimental situation. And they haven't been able to duplicate that in the field. The Deguche study is a field study, so it's probably has a lot of power for explaining things.
Our bacterial endosymbionts, has there been any evidence to suggest that bacterial endosymbionts could be associated with unpalatability in sponges?
With
unpalatability.
Oh, with the palatability, right. So one thing we do know is that some of the symbionts inside the sponges do make the natural products that are responsible for the palatability, right? So there's a group of Dicidia and lamellar Dicidia that have these oscillatoria symbionts, and we've actually shown that. We've figured out the gene cluster that codes for the metabolites, the secondary metabolites, and shown that that's actually in the symbionts and it's not in the host tissue, right? So we know that some of these do make the defensive metabolites and the things that make these unpalatable to fishes or turtles or whatnot, right? So, but that's only been done for a couple of species, and so when we start getting into this idea of how do we look at 1,000 host species, I don't know if the same mechanism is going to work the same way for every host, right? Right. Yeah.
Many of the questions lead to host factors, and whether there is any consideration of those host factors, how they control the internal community, and even within the species that were so many diversity. Is there anything obvious?
Right. So there's a couple of studies out, just coming out right now, that are on like toll receptors and showing how the receptors on the surfaces of the sponge cells are interacting with receptors on the surfaces of the symbiont cells, right? But again, there's only maybe 3 or 4 lab groups working on that area as well. But we don't have a general framework for that yet. So there's a lot still to do with the system. So if any of y'all are interested in more on that, come see me. And there's just a lot of big ideas that we still have to test and try to figure out.
Well, I think we can continue this discussion over pizza, and we can thank you all for.
Where are the tools for your tools today? Look in my lab, which is
yeah, hopefully. I think, what room is that? Yeah, are you going to send it to the other two?
So one of the SoMAS people is coming with them.